Hello everyone and welcome to ITLS Academy Empower the Youth. So today in this topic we will going to study about how to examine the microbial flora of vegetables and fruits. So it is a type of examination technique, means in this we will going to study through experiment and we will find what is the rate of spoilage in vegetables and fruits. So it is a simple food microbial analysis just to examine microbial flora of vegetables and fruits. Now let's quickly understand the introduction part. So we all are aware that microorganisms are always associated with plants and with animals. Means whatever is there in the universe, microorganisms are always present. They are so small that you cannot see with your naked eyes. So you need a special type of devices like microscope to visualize the presence of microorganisms. They are so harmful to humans, to plants, to animals, so such that they deteriorate the quality of each plant. If they are present in the food of animal, so they can do spoilage to them. If they are present in food of human beings, then also they causes different types of diseases. We all are aware that microorganisms are very much harmful for the human beings as well as for animals, for plants, if they are present. But there are also certain types of microorganisms that are not harmful, they are beneficial. For example, bacteria. Bacteria act as a beneficiary to humans as well as they act as a harmful to humans. So that is the reason microorganisms, if we talk about only the spoilage or if you talk about their harmful nature, so they deteriorate the quality of certain things. For example, they deteriorate the quality of food, make them unacceptable for the consumption. If they are present in water, they contaminate the water and make it fit for the human consumption to drink that water. So you should always treat the water before you consume the water. So they play a very important role for the survival of plants and animal. But on the other hand, growth of certain microorganisms are harmful. That I have already told you, that sometimes they are very harmful, but at the same time sometimes they are very helpful also. Microorganism in plant can result in spoilage, and sometimes they causes several types of diseases on consumption of such food. So when microorganisms, if they are present in food, so they cause spoilage in the food, and sometimes they causes various types of foodborne illness, means they causes various types of diseases on consumption of such food, means a food that have the presence of microorganism. So food spoilage by microorganism is due to increase in their number utilizing the nutrient, causing enzymatic changes, resulting in bad flavors due to the breakdown of some food materials or synthesis of new compound. Due to such microbial activities, food becomes unfit for human consumption. Also, food act as a good medium for transmission of many diseases. So this food is spoilage, so the food that is spoiled by microorganisms, so there are various type of reasons why they causes spoilage in the food. The first is, if due to the increase in their number, means microorganism, they have a capability of multiplying very fast, so they multiply very fast, and if their number increases, they contaminate the food particles, or you can say simply they deteriorate the quality of food in such a way that it becomes very unfit for the human consumption to consume or have that particular food. Okay, now utilizing the nutrient. So microorganisms also need a different types of nutrients for their survival, means they need food, and the food they get is from the food, from the nutrients that are present in the food. So they also, they also need nutrient for their survival, so they utilize the nutrients, so that they get, when they, that is the reason the food get deteriorated. Now comes the third reason, that is causing enzymatic changes that results in the bad flavors. So what they do, they cause the enzymatic change, means the enzymes get changed, and when the enzymatic changes occurs, means then it refers to the bad flavors, and this bad flavor is of course due to the breakdown of some food materials or synthesis of new compounds. So due to such microbial activities, food becomes unfit for human consumption, and also food act as a good medium for transmission of various type of diseases. Now comes this is a chart that shows that there are certain types of microbial defects of vegetables. So if you see, the first one is our beans. So if you see the image, the beans have certain types of spoilage that is occurring on the surface of their skin. So the defect is they have anthracnose, and they have the blit. What is the organism that is causing? The first is the Calectotriums, and second is a Xanthomonas. Second is the carrot. If you see the surface of the carrot, then there is a blackish type of thing that is present on the carrot. So this is because of the defect is the soft rot, or you can say fungal rot. Sometimes it is due to the decay wet, and sometimes it is due to the wet rot. So what is the organism that is leading to this type of defect? Then the first is rhizopus stolonifer, and second is the furicimps. Then the third is your onions. If you see the image of the onion, then this type of spoilage or defect take place. So that is because of the neck rot, black mold, and because of the Spurgillus niger or Pseudomonas species or Bilateral alii. Then comes the potatoes. So potatoes surface also get spoiled. So the first reason is because of the ring rot. Second is your dry rot, and due to because of the cyanobacterium and furicum. And the last is your tomatoes. So the skin of the tomatoes get spoiled. So just because of the different types of aspergillus or xanthomonas species and canidia species, they causes spoilage or defect. For example, like ferment, fungal rot, bacterial spot, soft rot. So these are certain types of defect that occurs in different types of vegetables. For example, they sometimes they changes the color of the fruits or vegetables. They make them dull. Sometimes the skin get easily very soft, and that softness is due to the microbial activity. Sometimes the surface get a little blackened in color. Sometimes there is a fungus grow. So all these occur, all these defect is due to the presence of microbial contamination. So these are certain reasons how the microorganism attack fruits and vegetable and make it unacceptable for human consumption. Now moving to the next, that is our experiment to understand this. So the basic aim behind the experiment is to examine the microbial flora of vegetables and fruits. So the theory says that fresh fruits and vegetables are exposed to potential contamination. It has been estimated that 20% of all the fruits and vegetables harvested for human consumptions are lost through microbial spoilage. The primary agent of microbial spoilage are bacteria, yeast, and mold. So if you see that different types of fresh fruits and vegetables, so they are exposed to the potential microbial contamination. Means the all that fresh vegetables, I'm not talking about the vegetables that are not fresh, so the only the fresh one I'm talking about. So the fresh vegetable, whenever they are majority of the fresh vegetables, they are exposed to the potential microbial contamination, and it is estimated through different types of studies that 20% of all the fruits and vegetables harvested for human consumptions are lost through microbial spoilage. So you can see up to how much extent this microbial contamination is exploiting the fruits and vegetables. That is 20% of all the fruits and vegetables that are harvested for human consumption. So the primary agent of microbial spoilage are bacteria, yeast and mold. So these three are the primarily agent, that is bacteria, yeast and mold. So the fruits and vegetables, so they carry the microbial flora which passing from farm to the tables. So of course the vegetables that are harvested in the farm, so they carry different types of microbial flora, and these microbial flora are passing from farms, means from the land where they are harvested, to the table. So the produce is exposed to the potential microbial contamination. So the produce, that is fruits and vegetables, so they are exposed to potential microbial contamination at every step. That includes cultivation or harvesting, transporting, the packaging, storage and selling to the final consumers. So if you see, then average consumption of fruits and vegetables, or you can say vegetables and fruit, is water is 88.3%, carbohydrate is 8.6%, protein is 2%, fat is 0.8% and ash is 0.3%. So it is an average consumption of fruits composition of fruits and vegetable, in which the highest content is water, that is 88.3. Next higher is your carbohydrates, that is 8.6. If you see the protein content, then it is 2%. Then of course if you see the fat content, then it is 0.8%, and if you see the ash, that is 0.3%. So the microbial spoilage and contaminating pathogen pose a serious problem in food safety as well as the economy. The examination of food is performed for the presence of a total number of bacteria in the food by SCP, and presence of coliforms for indicating the presence of pathogens. So if you see, if you see that microbial spoilage, means the spoilage in fruits and vegetables that are caused by different types of pathogen, that is microbial pathogens. So they are just, what they are doing, they are contaminating the fruits and vegetable, and this contamination is leading to a very serious problem whenever a food safety is concerned. And of course it is giving a loss to the farmers also, because their produce, means their products, that is fruits and vegetables, they are contaminating and getting spoiled through these contaminated types of bacteria, yeast and mold. So in this also farmers are getting losses, and of course we as a consumer are not getting the fresh fruits and vegetables. Okay, sometimes that is the reason why the cost of certain fruits and vegetable get increased, just because of the spoilage. Means sometimes the spoilage is up to which extent that majority of the fruits and vegetable get spoiled, only a small portion is left. So to distribute that small portion into the market is a big challenge, and that is the reason sometimes the vegetables get a quick higher rate than it should be. So it is again, sorry, the one of the way and this microbial spoilage, of course, it is a very serious problem whenever the food safety is concerned. That is the reason there are different types of regulatory bodies also, like GMP, like HACCP, different types of FDA bodies are there just to check the quality of the food that is going to be used by the consumer. Means as the a person that is going to consume that food. So it is a challenge for all the food industry to to dispatch those fruits and vegetables that should be of a high quality. Now, in this particular experiment, we need certain types of materials, that is collection of the food sample. So here the spoiled food samples such as vegetables, fruits, even the breads, they are collected from the local market area where they are used in the present study. Then we have the examination of spoiled food sample, how you examine them. So each food sample were examined very carefully for the spoilage symptom, like such as color, consistency, rot, odor, and other types of visible symptoms. So whenever when here one question arises, then that when you say that yes, now the food is spoiled, so there are different types of factors that lead to the spoilage. First, you check the quality of the food, like you check the symptoms, like whether the color has changed or not, what is the consistency, is there any rot attack, or if there is any a bad odor is coming from that particular fruits or vegetable. So these are certain visible symptoms that you check whenever you are purchasing any fruits and vegetables. In our present study, we examine the five things, that is pea, or you can say peas, tomatoes, apple, beans, and potatoes. So these are four things that we will going to examine today, and we will see the microbial spoilage in these five fruits and vegetables. Now, what is the procedure to examine? So the first is the direct microscopic examination of the spoiled foods, that is, after examination of spoiled foods for the symptoms, smear were prepared from the spoiled portion of the food sample and were examined under the microscope for identification of spoilage caused by bacteria and fungi respectively. So you take a sample of the spoiled product, and after taking the sample of the spoiled product, you make a smear out of it, means out of that spoiled portion of the food, or you can say of the fruits and vegetables. And after them, what you do, you examine the smear, or you can say you put that smear under the microscope, and you visualize, or you can say you identify the spoilage that is caused by bacteria, fungi respectively. So for the identification of bacteria, gram staining method were employed. So if you want to check for the bacteria, means if you want to identify bacteria, then you use a gram staining technique, that is gram positive, or simply you say that you take a gram staining method for identification of bacteria. So in this gram staining, in this the prepared smear, means the smear that is prepared was air dried, and they are heat fixed. Means what you do, you take that smear and you let it dry, and after drying you use heat to fix that smear. Then afterward, in the first step, it was treated with the crystal violet. So after that, you take crystal violet and you treat the fixed or dried smear for 1 minute. After that, you take distilled water and rinse that smear with the distilled water. Then again you treat it with the smear with the gram iodine, and you do this means after that, you when you are treating with a gram iodine, so you what you do, you treat it for 1 minute, then again you rinsed with water, and at last you take 70% of alcohol, and of course you rinse, you put it the alcohol on the smear, and you wait for 10 seconds. After that again, you take saffronine for 1 minute, and at last you washed it. After using these all three, you blot dried and observed under the oil emulsion, and for identification of the fungi. This is in the case with the bacteria. Whenever you are treating the whenever you are identifying the presence of bacteria, then comes the identification of fungi. So for the identification of fungi, the smear prepared was strained with the lactophenol cotton blue straining procedure, and you are observed under the 45x lens of the microscope. So if you see after the procedure, if you see the result, so the result is shown here. The spoiled food samples were then analyzed for different symptoms of spoilage, that is the figure 1 and figure 2. So if you see the figure 1, that is these, if you see this vegetables, so if you see it, the all the vegetables are spoiled, means if you see the upper, that is pear, so again this pear is having a patches of brown and black color, so so this patches indicate that it is spoiled. Similar case with the apple. If you see this area, again this area showing that this apple is not unfit for consumption. Even the skin become a little loose. Then similar case is with beans and peas. So this indicate that yes, the food is spoiled. And now moving to the next image, that is it shows the image of the onions and potatoes. So of course you can easily visualize that yes, this onion and this potatoes are unfit for human consumption. So what are the symptoms? If you see that figure 1 tomatoes, so the symptom is soft rotten tissue with a black rot, if you can see this image, right? Now second says tomatoes, so they have a large black rod. Then again you see the another image of the tomatoes, then there is a grayish black mold growth. Then with peas also, they have a black rotten tissues. Apple, they have a black spot on the surface. Same case is with green peas, so they have a black rotten areas on the surface. Again if you see the black beans, so they have a gray enlarged peas. And if you see the onion, they have a black powdery mass. And potatoes, they have a brownish color black color combination coloration with rot areas. So these are the food sample that you want to examine, and these are the symptoms of spoilage. Now if you see what is the microscopic examination of the spoiled food items or samples, so whenever you take any food sample, so you do a microscopic examination. So what does it means? Tomatoes, they have the presence of bacteria, that is gram negative rods. If you see the another image, they have a gram positive rods. And if you see the third image, that they have a fungus, that is mycelial filaments with bunch of spores. Similar cases with the beans also, they have gram positive rods with endospore. Then the beans, they have yeast oval shape. So again these are certain food samples, and they have a micro morphology of different types of morphology. Now moving to the next, that is the end. And let's see what we have studied in this experiment. So let's see the discussion part. So upon examination of the spoiled food samples, we can conclude that the spoilage is detected by visuals or external symptoms of spoilage of the food samples. These symptoms of the food spoilage are very several types, and consecutive organisms are spoiled. So in this, if you see that we have done the examination both by using the machine, that is microscope, and again by the external symptoms, like how the skin is looking like, is there any rot patches is there or not, what is the odor, what how the texture get changes. So this is again means here the external as well as visual symptoms of spoilage are considering. Then the type of spoilage is influenced by several factors. Means there is again there are not always the case that microorganism can only cause spoilage. Sometimes there is a chemical composition that can act as a spoilage factors in different types of food. They have a contamination of course by different types of microorganisms. Sometimes when there is a pH change, when there is a oxygen availability, temperature of storage, seas and moisture. So these are again there these are certain factors that is affecting the contamination, or you can say spoilage of different types of fruits and vegetables. So the symptoms observed were very for various types were said to be rot, softness, missiness of tissues, mold, slimy, souring, tint of flavors, of odors, spot. However, food sample is observed by direct microscopic examination. Wide varieties of organisms were observed, which depends on the type of spoilage. So, in our observation table, we could observe that gram positive rod shaped endospore forming bacteria and non endospore forming bacteria. So in addition, gram positive cocci in the bunches were as well as the single isolated cells or in pairs were observed. In case of spoiled beans, tomatoes, gram gram negative and rod bacteria shaped bacteria were observed, and wherein the case of the mold spoilage and the wide variety of fungi were identified. So now that comes to the end of the lecture, and let us understand the conclusion part. So such spoilage are mainly due to the contamination of various sources, such as during the harvesting of the crop, handlers, transportation, storage area, shelf life of the food product, etc. These spoilage can be prevented by prevention of contamination, by adopting preservation methods such as cleanliness at all the steps. So again, if you want to make your food item away from, or if you want to prevent the contamination of by microorganism, so it is always, you should always follow the GMPs, the different types of regulatory guidelines that says that cleanliness is one of the major step that you should always take whenever you are handling with food items. Because if the environment, if the storage condition is if the storage area is not clean, so of course there is a huge attack of microorganisms. So of course you should always take cleanliness as a very important step whenever you are handling the food with handling the food. So that brings to the end of the lecture. I hope you have understood this experiment. And again students, if you have any doubt, any queries related to the lecture, so you can simply leave your queries in the comment section below, and in the next lecture I will be answering the queries. Follow ITLS Academy on Instagram, Facebook, Twitter, YouTube, and WhatsApp, LinkedIn. And you can subscribe our channel of YouTube, that is ITLS Academy. You can also visit our website, that is www.itlsacademy.com. This is our helpline number, that is 7080833450. You can directly call on this number and get updated if you have any doubt and any queries. Again, thank you for watching. Please don't forget to like, share and subscribe. And yes student, please press the bell icon to get more update on different topics lectures. Thank you.